Dear viewers, welcome to the new lesson from Dr. Arts Biology. In this lesson, we are going to discuss about peptide bond formation and properties, in addition to which one tricky question also will be discussed and how to solve that particular question. We know that peptide bonds are formed between amino acids for the formation of proteins. So first of all, we need to know what is meant by an amino acid and what is the structure of amino acid. We know that amino acids are composed of a central carbon that is called alpha carbon, whose valence is filled by 4 different atoms or groups, except in glycine, where in glycine 2 of the valence is filled by the same atom called hydrogen. So let us see the structure of amino acid. First of all, this is the alpha carbon, and this alpha carbon 4 valence is filled by 4 different atoms or molecules. So let us see, the first one is nothing but the hydrogen. Second one, that is the amino group. It can be NH2 or NH3 plus according to the pH in which the amino acid exists. We will be discussing those things later in amino acid lesson in detail. So this is the amino group. Now, due to the presence of this particular amino group there, it will be known as the amino terminal. That end of the amino acid is known as the amino terminal. Now, next one is the carboxyl group. This carboxylic acid group will be exist in COO minus or COOH position according to the pH in which the amino acid exists. In acidic pH, it will be existing in the form of COOH, and in alkaline pH, it will be existing in the form of COO minus. And due to the presence of the carboxylic group at this particular end of the amino acid is known as the carboxyl terminal or C terminal or carboxy terminal. Now, the last valency will be filled by the side chain called R group. So this side chain will be varying among different amino acids, and that's why there are more than 20 different amino acids possible. So the R group will be filled in the same H in case of the glycine. That's why I told in the first part of this lesson. Now let us see the formation of peptide bond. We know that peptide bond is a covalent bond between two amino acids in a protein or peptide formed by the elimination of one water molecule. Let us see how it will be formed. So one amino acid, now it will be combining with the second amino acid. We can see I am just putting R1 for the first amino acid and R2 for the second amino acid to represent the side chains. Now it will be forming a peptide. This peptide is known as a dipeptide because it is formed of two amino acids linked through a peptide bond. During this formation, there will be a concomitant release of water molecule. This water molecule is nothing but the hydroxyl group of carboxylic acid group in the first amino acid and hydrogen atom that is attached to the nitrogen of the amino group of the second amino acid. So that is what this particular water is. Now, as a result of this loss of hydroxyl group and hydrogen from carboxylic acid and amino group respectively, there remains one valency left for the carbon and nitrogen. This valency will be filled each other by sharing of electrons, and that results in the formation of this double bond. So this particular carbon will be having one valency left. Now this nitrogen also will be having one valency left. As a result, they will be sharing the electrons, and as a result, the covalent bond, peptide bond, will be formed. As I told you before, in a peptide, the end which contains the amino group will be known as the amino terminal, and the end which contains the carboxylic acid group will be known as the carboxy terminal. Now let us see the properties of peptide bond. So we have to remember the first that a peptide bond is a bond which results in the formation of different proteins, and it is formed between the carboxyl group of one amino acid and the amino group of the second amino acid, just like here with the concomitant formation of the water. Now the second property is the different lifetime of a peptide bond in aqueous solution. The absence of a catalyst approaches 1000 years. So it is one of the persisting bond in the absence of catalyst. Catalysts are nothing but the enzymes. In the absence of enzymes, or in the absence of any catalyst other than enzyme in an aqueous medium, the peptide bond will be surviving more than 1000 years. But we can see one contradictory fact that the equilibrium of the reaction, that is the equilibrium of the reaction that is given in this particular figure that I represented above. The peptide bond is formed, in which the peptide bond is formed, lies on the side of hydrolysis. Hydrolysis means the breaking of bond. So you can see the new yellow arrow mark. So equilibrium is towards the breaking of bond, not towards the formation of bond. But still the peptide bond is persisting. The reason is the stability of this peptide bond is actually resulting from the fact that hydrolysis takes place very slowly in the absence of catalyst. So keep in mind the equilibrium of the reaction in which the peptide bond is formed is actually towards the breaking of peptide bond. Still in this condition, the peptide bond is more stable because the hydrolysis takes place very slowly in the absence of catalyst. This particular point can be asked as a tricky question in competitive examination. Now next property is almost all peptide bonds in proteins are transposition or transconfiguration. This is just to avoid steric interaction or steric clash that will be or that would happen in cis configuration. You don't get confused with the trans and cis configuration. It is easy to remember. Relate cis word with the same, as the first position, first letter is almost pronouncing same, cis same. You can just remember in this way. So cis same, trans is different. So cis will be having the atoms or molecules organized on either side in the same position. So you can see here in this C, in this cis position, so the oxygen and the hydrogen on this peptide bond is actually on the same position, and also the R group of the first amino acid and R group of the second amino acid is also in the same position. This will be resulting in steric clashes that is marked here. So in order to avoid this steric clashes, the atoms or molecules are organized in trans configuration like in this figure. So we can see here the oxygen of the carboxyl group and hydrogen of this one is in opposite direction at the peptide bond, and also the side chain of the first amino acid and the side chain of the second amino acid are in opposite direction. This particular configuration is known as the trans configuration. So almost all peptide bonds in protein will be in trans configuration. So keep that in mind. It is one of the multiple choice question that will be coming during competitive examinations. You can also just look at this particular website. It is giving some the bond angle and all description. You can just visit that site. The next property is peptide bond behaves like a double bond, even though we are representing it in a single bond, or chemically it is a single bond. It will be behaving like a strong double bond that is restricting the rotation at this particular bond. So peptide bond behaves like a double bond, and therefore free rotation at the bond is strongly restricted. But even then, we know that proteins are forming numerous folds, and that's why it's attaining the three-dimensional and quaternary structures. How it's possible? The reason is the strong single bond nature of the bond, that is important bond, that is present in the amino acids. Let us see what are that particular bonds. We know that this is the alpha carbon, and this alpha carbon is linked to amino group, and that bond is known as the phi bond. This phi bond is one of the main factor by which proteins can or peptides can fold in any extent. Now, second valency of the alpha carbon is filled with the carboxylic acid group, and that bond is known as the psi bond. This is the second bond that is important for folding property of the amino acid, proteins. Now, there is a peptide bond. So, we can just see that there is the remaining valency will be filled by the R group and hydrogen. It is not having any involvement in the free rotation possibility of the polypeptides. So, that's hydrogen and side chain is there. So, you can see the peptide bond is also there, that is linking the amino acid in a polypeptide, and this peptide bond is otherwise known as the omega bond. So, omega bond, even though it is single bond in nature, it is having a strong double bond nature that is restricting the free rotation, but this restriction is overcome by polypeptides due to the high degree of single bond nature of the phi bond, which is connecting alpha carbon with the amino group, and psi bond, that is connecting alpha carbon with the carboxylic acid group. So, due to psi and phi, the free rotation is possible, and therefore free folding of polypeptides are possible in order to attain different forms of structures. So, you can see the psi bond, you can see the rotation that is given here, and the psi bond. So, this one is a phi bond rotation, and this one is the psi bond rotation that is given. You can just see that. For getting more figures like this, you can just visit the website Quizlet and you can just look into that. Now, let us see how this present session helped you. Let us see some question. So, consider the question. The flexibility of polypeptide even at the restriction due to strong double bond nature of peptide bond is favored by the strong single bond nature of bonds except. So, options given are psi bond, phi bond, omega bond, it is not the om bond, omega bond, and then both B and C. So, we know from the previous slide that the answer is the omega bond. C is the answer because the phi bond is helping in free rotation, that is a bond between amino and alpha carbon, and the psi bond is also helping in free rotation, that is a bond between alpha carbon and carboxylate group, and this omega is the bond between the peptide bond between two amino acids, that is actually of strong double bond nature, and that is restricting the free rotation. So, option C is the answer for this particular question. So, this kinds of tricky question will be coming from the peptide bond, and more details of peptide bond and protein structure formation will be there in the upcoming videos. Please wait for that. Thank you for watching this video. Hope it's helpful for you, and you can just share to reach your friends. Also, don't forget to subscribe my channel and click on the bell button in order to get the notification when I am uploading the new videos. Thank you.